Hello everyone, welcome to my channel Life Sciences Tutorials. I am Nishi and I will be guiding you through all the important topics which are required for your various college examinations and also for various competitive exams. If you want to go for higher level studies, or maybe you want to do PhD, you want to do research, then there are so many competitive exams, and to crack those competitive exams you need to study in a different way. So I will be guiding you to the various topics, all the important points which one should remember while preparing for the competitive exams. I will specially deal with biology or life sciences. So I think all of you are aware of the CSIR NET examination. So there are different subjects for this, and the one which I am dealing with is life sciences. Now, when we talk about life sciences, it's a very broad term. It includes the entire biology and all the related fields. So it's very difficult to study everything which is mentioned in the syllabus of CSIR NET examination. And if you prepare for CSIR NET, you are like already prepared for all other, maybe specific university entrance exam or any specific college entrance exam. So the syllabus includes everything related to life science. When students are actually studying, maybe during the graduation or post graduation, we do our specialization in a specific subject, and we study a specific set of subjects. Like one might be doing their masters in botany or zoology or microbiology or biotechnology, and there is no field wherein you literally study everything which is mentioned in the syllabus of life sciences. Now, we do touch upon different aspects of each unit, but as such, the syllabus, if you see this, the syllabus is so broad that one cannot study each and everything in just one degree. But when you are trying to give this exam or other college entrance exams, then all this becomes very basic. So first, let's get introduced to the syllabus for CSIR UGC NET examination, life sciences field. So as you can see, these are the various units which are part of the entire syllabus. So first, let's quickly get familiarized with the entire syllabus. Then in the upcoming videos, I will teach you in a very simple way how to understand all the different important topics in life sciences. Okay, so first let's see all the units. So we have molecules and their interaction relevant to biology, cellular organization, so this is simply the cell biology. Next, we have fundamental processes, so this includes various processes which we study as central dogma, like replication, transcription, translation, its regulation, very important unit. Then we have cell communication and cell signaling. We have developmental biology, we have system physiology. First we have plant, and then we have animal. Then we have inheritance biology. Next, diversity of life forms, ecological principles, evolution and behavior, applied biology, and methods in biology. So these are the various fields, many of the topics which are distributed in all these 13 units. Some of these we study maybe in a certain subject during our graduation or post graduation. Then there are many things, especially here in the methods of biology, which we mainly study in the practicals during our graduation and post graduation. So it's like some total of all things which you study and a little bit more. So these are just the titles. We will also see what are the topics which are mentioned is in each unit. But definitely there is no end to this syllabus. If you are aiming for higher education, so it is expected, like you know all the basics. So don't be intimidated that how will I study so much. If you channelize and if you strategize everything properly, I think it is very easy to prepare it in a few months. And rather, I would say during your post graduation itself, like during the 2 years, if you study all the content properly, then I think you are already half prepared for the competitive exams. Now, the nature of competitive exam, like here also we have the multiple choice questions wherein you have one single correct answer. So I will also tell you about the marking scheme, the paper pattern in the next video. But obviously you have MCQs and you have single correct answer. So, and there is negative marking. So one must do the right choice, and you should have accuracy plus speed. So it is important that you prepare for the competitive exams accordingly. It is not only about knowing the concepts, but it is also about how you are able to apply those concepts. This exam is mainly for you getting into research, like if you want to do PhD. So to do PhD from reputed national level institutes, you need to crack this exam. And also if you want to teach college students, then this is required for the lectureship. So for this, definitely you should be acquainted with all the topics. Now quickly let's go through all the subtopics or the topics which are included in each unit. Now we will just go through, I will read out everything so that you're familiar with the entire syllabus. Don't worry, obviously no one can study literally everything from every unit, but if you focus on specific units depending on your specialization, obviously if suppose you have studied botany, then you will be very familiar with the system physiology, plants, then also biochemistry, some basics of fundamental biology, right, some of the methods also. So everyone has studied much of it, but when you put together everything, it becomes like, oh my god, how we are going to study. Let's start with the first unit. So we have first unit as molecules and their interaction relevant to biology. So first we have structure of atoms, molecules, chemical bonds. So you can understand we are talking about very basic stuffs, basic chemistry, which you start studying from your school. What are atoms, molecules, different types of bonds, and then only you'll be understanding, the different kinds of biomolecules. So definitely carbohydrates, lipids, proteins, nucleic acids, and vitamins, their structures, then the different kinds of stabilizing interactions, the different kinds of bonds which help to stabilize the different levels of structures, then a little bit of biophysical chemistry. So definitely, if you go through the previous year question, you will get many questions on pH, buffer, such topics, reaction kinetics. So there is also a topic on enzymes. So enzyme kinetics, it's like a favorite topic of the paper setters. Next, we have a little bit of thermodynamics, colligative properties, and very important, we have bioenergetics. So different metabolic pathways here. We have glycolysis, oxidative phosphorylation, coupled reaction, group transfer, biological energy transducers. Then we have the principles of catalysis. So definitely the enzymes part, very important. As I said earlier, enzyme kinetics, regulation, mechanism of enzyme catalysis, isoenzymes, very important. Then we have the conformation of proteins, very important topic. We have Ramachandran plot, then the secondary structure, domains, motif, folds, right? So different levels of protein structure. Then we have nucleic acids, the different forms of nucleic acids, right? Different forms of DNA. Then we have the different types of RNA, right? Very important. DNA and RNA, both their structure, functioning. Then we have the stability of proteins and nucleic acids, and basic other, metabolic pathways, that is metabolism of carbohydrates, lipids, amino acid, nucleotides, and vitamins. Next, we have the second unit, that is cellular organization. So this is simply the cell biology part. So obviously this is very easy. We have the different parts of cell structure. So first we have the membrane structure and function. So the lipid bilayer, different models, how there has been evolution in our understanding of different models of plasma membrane, right? Then the transport, see here the different types of transport. We have the membrane pumps, mechanisms of sorting, regulation of intracellular transport, electrical properties of the membranes. Then we have the organelles, right? Very important, starting from all the different parts of cells. So cell wall, nucleus, mitochondria, Golgi bodies, lysosomes, endoplasmic reticulum, peroxisome, plastids, vacuoles, chloroplast, structure and function of cytoskeleton and its role in motility. Next, we have the organization of genes and chromosomes, obviously very important parts of the cell. So here we have included again a little deep concepts like what is operon, unique and repetitive DNA, interrupted genes, gene families, structure of chromatin, chromosomes, heterochromatin, euchromatin, transposons. Obviously, many of things we start studying from our 11th, 12th itself, like what are genes, what is chromatin, its packaging, chromosomes. And if you're familiar, if you're thorough with all these concepts, then it is very easier to understand other topics also, like how cell divides. Next, see the next topic, we have cell division, cell cycle, so mitosis, meiosis, its regulation, the different steps, control points, checkpoints. There are many previous year questions on checkpoints. Next, we have microbial physiology, so bacterial and fungal related concepts. So we have the growth, yield, characteristics, strategies of cell division, and stress response. So this was the second unit. Next, we have the fundamental processes. So in fundamental processes unit, very basic, everything related to central dogma of life. So here we include topics like DNA replication, repair, and recombination. This is very important unit. Rather, I would say this is a unit like wherein we can easily demarcate, like where to stop. And if you study from like good standard books, genes, maybe Nelson and Cox Leninger, right? Biochemistry, then this will be very easy for you. So unit of replication, what are the enzymes involved, origin, replication, for all the details of replication. Then if there is any damage, so any mutation, then how to repair it, the different mechanisms, then homologous and site-specific recombination. Next, we do have RNA synthesis, that is transcription. So everything related to transcription factors, machinery, the entire process, initiation, elongation, termination, all the steps. RNA polymerase is very important. It's processing. Like in eukaryotes, we have processing like capping. We do have other steps like we have tailing. We have different steps like elongation, termination, RNA processing. You can see RNA editing, splicing, polyadenylation, that is tailing. Then the different types of RNA transport. Next, we have protein synthesis. So the translation process. To understand translation, we should understand the different types and structure of ribosomes, right? Then the everything, all the steps involved in translation, tRNA, the genetic code, right? And further post-translational modifications. Next, we have control of gene expression at transcription and translational level. So obviously here you will require the concept of operon also, wherein we understand the concept of gene expression, its regulation. Apart from this, regulating the expression of phages, viruses, prokaryotic and eukaryotic genes, gene silencing, another important topic. This is very, upcoming field. There is so much research done in gene silencing. That's a huge part of biotechnology. Next, we have the cell communication and cell signaling. Very important. This is a big unit. If you go through previous year's questions, then I think you can easily figure out, the topics which are very crucial for competitive exams. So in the upcoming videos, I will guide you and I will teach you like which are those topics, and like those are do or die topics. You have to do it. Okay, like your cancer and some of the topics, like you don't just have to look at is it important for exam or not. If you're interested, go on, read more about it. And for research purpose, definitely, these topics are important, especially cancer, immunity, you should be aware of it. So let's go through this unit. We have the topics like host-parasite interaction. So recognition and entry response of different pathogens, very important. Okay, bacteria, viruses, animal and plant host cells, alteration of host cell behavior by pathogens, virus-induced cell transformation, very important. Pathogen-induced diseases. So just get acquainted with various bacterial, fungal, animal, plant diseases. Cell fusion, in both normal and abnormal cell. Then we have very important cell signaling topic. So your hormones and the receptors. Next, the various signaling through G coupled receptors. Very important. This is important. Signal transduction pathways, second messengers, then the bacterial implant, two component system, light signaling implants, bacterial chemotaxis, and colon sensing. So we'll touch upon all the important topics here. I mean, cellular communication. So simple, this includes all the junctions, okay, how transport occurs and all those things. Extracellular matrix, integrins, neurotransmission, very important. Next, super important, cancer topic. So genetic rearrangements in progenitor cells, oncogenes, tumor suppressor genes. So different kinds of genes which are related to cancer and tumor. Next, how it is related to cell cycle, virus-induced cancer, very important property, metastasis, interaction of the cancer and normal cells, apoptosis, very important. There are many PYQs on this. Then therapeutic interventions of uncontrolled cell growth, very important. Next, we have the innate and adaptive immunity. So entire immunology, in college we study it as, a separate paper, but here it is just a topic. So obviously it's like, oh my God, how will I remember everything? So go through all the PYQs and you'll figure out, okay, these are certain things which you have to study. And if you want to pursue your career in this field, then definitely study everything. The standard book for this will be obviously, my favorite is QB Immunology. That's the best book to study immunology. So all the topics about innate and adaptive immunity, what are antigens, antigenicity, different properties, immunogenicity, then everything about the B and T cells, antibody structures, the different kinds of WBCs, then obviously other related topics like inflammation, hypersensitivity, autoimmunity, immune response during various diseases. It could be bacterial, parasitic, viral diseases, right? Congenital diseases, so genetic disorders, innate, and then acquired immunodeficiencies, vaccines, very important. Okay, next we have unit. Okay, now this is one unit which not everyone has studied during their college. Some of you, depending on your degree, you study, and there are many degrees wherein actually you don't have this subject. So then for those students, this becomes like a big challenge. How will I study? But trust me, if you're interested, you can target this. Another important unit, there are many questions which are asked from this unit. And actually, if you study, you will understand those are pretty straightforward questions. Many of the questions are repeated on, like same topics. And a very interesting unit, developmental biology, very important. So I will tell you all the topics now, but if you study properly, use a good book, like you can use Gilbert's, okay, and then you're sorted. So basic concepts of development. So we have, these are like basic terms, okay, potency, commitment, this is with respect to cell. Next, we have specification, induction, competence, determination, differentiation, like different stages in the development of cell. Next, morphogenetic gradients, cell fate and cell lineages, stem cells, very important, genomic equivalence, cytoplasmic determinants, imprinting, mutants, transgenics and analysis, very important. Then various processes, like right from, fertilization, we have zygote, and then we have embryo, fetus, how this development occurs. So during this, we have development, differentiation. So all those steps are included here. Okay. Again, here both plants as well as animals are included. Okay, it's not only plants. As you can see here in this particular unit, it's everything mostly related to plants, which is given, and then we have animals also. Next, we have morphogenesis and organogenesis in animals. So these are some classic cases to understand the different concepts related to developmental biology. And study this, and you will be easily able to solve one or two questions in every exam. Okay, so very important. So these are some, like model organisms in which developmental biology has been studied. So very important. Cell aggregation, differentiation in dictyostelium. Then we have axis and pattern formation in Drosophila, very important topic. Okay, amphibia and chick. Then we have organogenesis, vulva development in Sinorhabditis elegans, C. elegans, very important topic. Eye lens induction, limb development, regeneration in vertebrates, differentiation of neurons, post-embryonic development, all these larval formation, metamorphosis, environmental regulation, sex determination, very important, easy topics. To study once, I think you're sorted. Next, we have morphogenesis and organogenesis in plants. So, study based on PYQs, then you will be sorted. Okay, so organization of shoot and root apical meristem, shoot and root development, leaf development and phyllotaxy. Then we have transition to flowering floral meristems. Then we have floral development in Arabidopsis and Anthurium, very important. So plant-specific developmental processes. Okay, and last we have the programmed cell death, apoptosis, programmed cell death, very important, aging and senescence, because that is also part of developmental biology. Next, I think this is pretty easy to guess what will be present in this unit. System physiology plant. So different components of plant biology and physiology, metabolic pathways, right? Plant hormones. So definitely all the details of photosynthesis, respiration and photorespiration, nitrogen metabolism, because obviously very important for plant growth. Then we have plant hormones, sensory photobiology. This is an important part of the plant physiology unit. Next, we have some other Remaining topics here you can see we have the solute transport and photoassimilate translocation. So right from uptake of minerals and water from soil to its transport, how xylem and phloem are involved in this. Then different concepts like transpiration and how it actually helps in the ascent of sand. Secondary metabolites. Important, remember roles of some of the important secondary metabolites and stress physiology, both biotic as well as abiotic stress and how plants respond to it. Next, we have system physiology, animals, I think animal physiology. So different systems in detail. Okay, so blood circulation, cardiovascular system, respiratory system, nervous system, sense organs, excretory system, thermoregulation, we have stress and adaptation, digestive system, endocrinology, and reproduction. These are all the important topics from animal physiology unit. Very important, especially stress on the endocrinology, digestion, cardiovascular, blood, respiratory, and maybe a little bit of excretory also. Some of the specific questions are also asked on sense organ, but I think if you focus mainly on these start topics, then most of the questions will be done. Next, my favorite, we have inheritance biology, simply genetics. Okay, so right from very basic, we have the Mendelian principles, the 3 laws given by Mendel, the concept of gene, what are alleles, different types of process, right? Extension of Mendelian principles, the exceptions which we study later, like codominance, incomplete dominance linkage, other topics like pleiotropy, genomic imprinting, sex linkage, very important. Next, we have gene mapping methods, very important. Okay, there are some questions on the molecular markers. Next, we have extra chromosomal inheritance, so mitochondrial, chloroplast, and maternal inheritance. We have microbial genetics, we have human genetics, so pedigree analysis, very important, a little tricky, but I think if you understand the genetics and how actually, the dominant, recessive, autosomal, sex-linked genes are inherited, I think you can easily solve any pedigree cross. Next, we have quantitative genetics, so it includes like topics like polygenic inheritance, QTL method. Next, mutation, very important. Okay, mutation types, what are the mutagenic agents, mutagenesis, insertional mutagenesis, very important. So obviously for biotechnology, also can relate it. Structural and numerical alterations of chromosomes. So here again, this is like extension of mutation only, but here we will talk about mainly the chromosomal mutations like deletion, duplication, translocation, inversion. Ploidy concept, very important. Next, recombination, homologous and non-homologous recombination. Next unit is diversity, very basic. You study this right from 11th class. I think even if you remember the details and the content from your 11th class textbook, it is pretty sufficient to solve most of the questions. So, do a very basic study of this. So, concept of like what is species, the hierarchy, right from kingdom, everything. Next, levels of organizational, structural organization. So, different, levels like unicellular, colonial, you may have all levels of organization, right from cellular, tissue, organ systems, very basic, symmetry and all those topics, adaptive radiation, little bit of evolution part is associated here. Next, outline classification of plants, animals, microorganisms. So their relation, how these organisms are classified, the criteria used, natural history of Indian subcontinent. So this is again basic study from some book which is specifically for competitive exam, like maybe Pathfinder and all, that will be sufficient for this. Next, organisms of health and agricultural importance. And last, we have organisms of conservation concern. Read a little bit about this also. So here we have the different categories for conservation, the strategies, and more about endangered, critically endangered, vulnerable, all these categories, okay, of species. Next unit, the 10th unit, we have ecology, ecological principles. Again, maybe basic school or 11th, 12th, whatever ecology we study, that knowledge. And based on the previous year questions, if you prepare, I think this is pretty easy unit. So one can prepare, and there are many questions which are asked on ecology unit. So I think even if you have not studied it, maybe in your college, but if you're targeting for any competitive exam, then this is good. Okay, so everything related to environment, habitat, and niche. The difference between it, population ecology. This is important. There are many questions asked on this. See, other topics you just read it. That is just to know the background of it. But whatever I'm marking, like the star mark one, this is important. Like there are many questions on R and K selected species. Okay, so you should know this. Next, species interaction, very important. Direct question. Community, ecology, even concept like ecotones. There are direct questions on this. Ecological succession. Read basic. This is pretty easy. Like 12th standard ecology part, that is sufficient. Next, ecosystem ecology, a little bit of energy flow, mineral cycling, cover some of the important biogeochemical cycles, that is sufficient. Different aspects of the ecosystem, okay, ecosystem structure and function in short, and biogeography. So this you will get again in some specific book for competitive exams, so focus on that and do this. Next, we have applied ecology, and then conservation biology. Read a little bit on this also. Reading I think is quite sufficient. Next, we have evolution and behavior. So very important. Again, this is a huge unit, but go through the topics, read it, and go through the PYQs. I think you will easily understand what the syllabus are. I'll tell you the entire scope of the syllabus. So here we have the emergence of evolutionary thoughts, different theories given all throughout. Next, origin of cells and unicellular evolution. So all the even the experiments, all the theories which were involved, right from prokaryotic, eukaryotic cells, complex life forms, the entire sequence. Next, paleontology and evolutionary history. Very important. There are many questions asked based on the evolutionary time scales or the eras and all. There are many questions. PYQs, so do it. And major events in the evolution. Like when fishes evolved, when was the golden age of fishes or amphibians and whatever some plants. Very important. That and these are direct questions. So it's like either you know or you don't know. So focus on that. Next, we have molecular evolution. Do a little bit of it because there are some questions on protein and nucleotide sequence analysis, genes. Okay. Next, we have mechanisms. So this will include the population genetics. Do cover this. Very important. Hardy-Weinberg law and all. Very easy and important to understand. Also, concepts like adaptive radiation, genetic drift. Very important. Or whatever I'm marking is important. Like there have been many questions asked on these topics. Direct questions. Speciation, allopatriacy, and sympatry. Okay. Very important. Next, convergent evolution, sexual selection, and coevolution. This part is important. Brain, behavior, and evolution. Maybe read it and focus on topics like, again, this skin selection, altruism. There are many questions on this. Next, see here, further, we have biological clocks, development of behavior. If you have studied animal behavior also, I think most of this part will be covered, but it's okay if you don't go very deep in this. If you're interested, you can read it. But solve PYQs, I think you will find out what to read. Okay, but do read it if you have time. And very important, don't skip this. I think whatever it is, whatever your goal, maybe you want to become a teacher in college, and so your main goal is to clear CSIR NET examination lectureship. Okay, then still as a teacher you should be very thorough with some of the basics, and as a research student or research scholar also, in future you should notice. So some basics on this unit. Again, there's no end to this, but do study about transgenic animals and plants, or study about cell culture. This is the future. You should know this if you are keen in research. A little bit of fermentation, though maybe direct questions may or may not be asked. There are not many questions, but still go through it, at least for some important, commercially important products. And this one, marker assisted selection, you should know these topics. Genomics, gene therapy. And last but not the least, super important. There are many students who ignore this, but I would say this is a very important unit. Again, I will repeat, if you want to become a teacher or you want to pursue research, then definitely, go through all the techniques. So right from molecular biology and recombinant biology, right from, isolating DNA, RNA, gel electrophoresis, separating DNA, blotting, cDNA libraries, what are plasmids, different vectors, DNA sequencing, like everything. Slowly, bit by bit, but do study it. There are many questions based on, you should know what's the concept or the principle behind the technique and how actually it's done, how to interpret a particular result, So based on those questions I asked. Then histochemical and immunotechniques, maybe we'll intro to this and get familiarized with these techniques. Very important. Next, we have biophysical methods, a little bit of spectroscopy, its different types, and statistical. Again, here for this, I would say if you've studied statistics, it will be easy. Otherwise, studying entire statistics or biostatistics will be difficult. So maybe focus on the important topics. Look at the PYQs. Focus on the important topics, and you will get some questions on the correlation, regression, and all. But I would say, though, many direct questions are not asked, but it's important for research. If you want to become a researcher, this is super important. So do study this. Like when you do the experiments, you will do all these t-tests, errors, and standard deviation. This is important for research. Last, here we have the radio labeling techniques, important, and this is pretty easy. Okay, you can do it in one day also. Next, microscopic techniques, a little bit of basics, the fundamentals of microscopy and different types. Next, electrophysiological methods. So a little bit of this medically important techniques like ECG, PET, MRI. Get familiarized. Though direct questions, there are not many direct questions on this, but for future, it's important. And methods in field biology, you also go to PYQ, so you'll get some standard questions on this and focus on that. Otherwise, if you've studied it already, maybe in your graduation or post-graduation, I think it will be easy for you to do this. Okay, a little bit of this. So this is the entire syllabus for CSIR NET examination. And as you can see, the syllabus itself is huge, right? So one cannot do it in just few days. So you need to give time. What I would tell you is, whatever you are studying for your college examinations, whatever, maybe your internal test or your semester and final year end test, whatever you're studying, study it thoroughly. Don't study it only for some exam purpose, because now we have seen the entire syllabus. It's very clear that whatever you have studied in your college, you have the same topics. Pretty much if you study well in your graduation and post graduation, mainly I would say, then definitely in those 2 years of post graduation, as you study all the regular, content of papers, you are half prepared for any competitive exam. Later on, slowly, bit by bit, get familiarized with the syllabus, with previous year questions, practice questions. Depending on your degree, that you will definitely have some topics which you don't study. See, enzymes, I think pretty much whatever you choose, everyone studies about enzymes. The extent may be different, but everyone has the topic. But if someone is doing specialization in botany, then the person might not be that good at animal physiology because he or she has not studied it, right? So definitely that time you have to give more time for that. And it is not that you have to study all the units. No. Okay, I've told you all the important points. Pick your interest, go through PYQs. See, clearing a competitive exam should, have a very solid different strategy, and it will also depend on what was your core subject, what was your specialization. Okay, like my specialization was biotechnology, so a greater part of genetics, like the inheritance biology, then even a bit of developmental biology, techniques, then enzymes, the basic starting units, fundamental processes, replication, all those was, pretty much covered during my studies. So now there were some things like which were not directly covered, like maybe ecology. I had some parts of ecology in my graduation program, but by the time you forget everything. So you know the basics, but you don't remember every bit of it to solve the questions in competitive exam. So you need to revise it, and then we'll be sorted. So I hope this tutorial was helpful for you. In the upcoming video lectures, I will teach all the important topics required for your regular college examinations and also for various competitive exams like CSIR NET. Till then, bye bye.